Hi everybody, it's a great pleasure to be here. So today is a split class in which we're going to have a first half which is theoretical. We're going to see everything interesting to know about repeats and genome assembly. And after the break, we're going to have a pretty long breakout group activity where you guys will have to assemble the genome and again get to practice on the cluster. It's going to be really good for your skills and also to help you for the midterm. So let's jump right into it. Most repeats are mobile DNA. So repeated sequences in a genome are sequences that are present multiple times. It's as simple as that. And they can be exactly identical or almost identical. So as long as they're kind of almost identical, it's counted as repeat. There is a bit of a fuzzy definition of what is identical enough, but mostly anything that's homologous and that looks similar enough would be considered a repeat. So there are two classes of repeats because there are two classes of mobile elements, of mobile DNA elements. There are actually more classes of repeats. And there are the mobile elements of class 1 and the mobile elements of class 2. So these are selfish elements, so DNA that are pretty small and they just make copies of themselves inside a genome. They don't really leave the genome. They're not autonomous in that sense. So they're somewhere between just a gene and a virus, something like that. So the two classes of mobile DNA are based on the mechanism of replication. Class 1 elements use a reverse transcriptase, class 2 elements use a transposase. We're going to start with class 1, and we know this enzyme, the reverse transcriptase, is the one that goes from RNA to DNA. So all the elements of class 1, and there's the LTR retroviruses and the non-LTR elements, produce an RNA, which is indicated in blue here. This RNA is exported, is translated in the cytoplasm, and then there is the class that directly translates, make it cDNA, and then goes back to the genome. Here, that's this class, the non-LTR. Just like it's a very short trip. RNA goes to the cytoplasm, protein, make a cDNA, go back to the genome. In reality, the cDNA is even produced inside the nucleus. And the other members, these ones, they're kind of weird. We're going to see this a little bit later. They produce an RNA, they go to the genome, translate it, they make a capsid, just like a virus. That capsid allows reverse transcription. The reverse transcription happens in that capsid instead of in the nucleus. And then that DNA is put back in the genome here. So it's kind of weird. Like they make a capsid inside the cytoplasm and they never leave the cell. So they make copies inside the genome. Class 2 don't have an RNA intermediate. They go directly from DNA to DNA. So it's always the same kind of logic, even though you have different enzymes that do this. But at the end of the day, that means that the DNA is kind of cut and pasted somewhere else. Okay, so you can have any of these three possible mechanisms, but at the end of the day, these two classes here, they have just moved. They haven't even made a copy of themselves, and this one has actually made a copy of itself because it actually used one strand to make a copy, the other strand remained in place, so at the initial site you still have the copy. So two classes of mobile elements, the one that have an RNA intermediate and that use the reverse transcriptase, the ones that don't have an RNA intermediate and use only DNA. If we look in the human genome, most of the repeats are one of these four things here. At least that's most of the mobile elements. And that's a slide that we've seen in the very first class when we described the human genome. So the lines here is by far the most abundant, and they're like 6 to 8 kbs in length. There is approximately 85,000 of them, so that's a large number. If you compare to the 20,000 genes, there are much more genes coming from these line ones here than from the human genome. And just that constitutes 21% of our genome. It's always the same sequence or very close all the time. Like this is a pretty big portion of the genome that corresponds to these repeats of 8 kb. Another category is the signs that are much shorter but much more abundant, 1.5 million of them. And they're like 1 to 300 base pairs. And they themselves represent 13% of the genome. So these two together combined correspond to 44-45% of the genome, so that's really most of the genome. You also have retroviruses like HIV, for instance, like you don't have HIV in the genome, but retroviruses like HIV. And there are a lot of copies, 450,000. That's even more than the number of lines. But it's because mostly they're broken. They have been, there are deletions. They have plenty of mutations, so they're shorter. And so they can be from 1.5 to 11 kb. That's 8% of the genome. So again, like much more than the protein coding content of our genome. And you have the transposons, the fossils. So things that don't really make copies of themselves, they rather move. Like these ones are class 1 and this is class 2 here. And these ones, it's kind of a mixed bag. You have plenty of things. Can be short, can be relatively large. There is a large number of them, 300,000, but they represent only 3% of the genome, mostly because they're kind of small. Yes, either Theodora tested.
Okay, Dr. Filio. So I have a question about retroviruses. So I know that, with this retrovirus, it can create more DNA sequences. So does this mean that our genome has the ability to expand itself in any way?
Sorry, the sound is not so good. Can you repeat that?
Oh, okay. I apologize. Can you hear me now?
Yeah.
Okay, great. So, given that retroviruses can make an mRNA copy of like a DNA sequence and then create a cDNA copy of that and put it back in the genome, does this mean that our genome has the ability to expand itself further? Can it have more nucleotide bases if it comes to that?
So again, like the sounds keep coming up and down. It's very difficult to understand you. But the question is, does it mean that our genome what? I understand the first part, but does it mean that the genome what? I couldn't understand.
Could it expand the past 3.2 billion base pairs?
I don't hear well. Like every time you say the word, then there's something that beeps out, and so I don't even understand the question. Maybe type it. I don't know. Can the genome extend? I think Margaret understood it. Can the genome extend? Yeah, of course the genome can grow. Like if this thing is happening quite fast and the genome grows. Thanks, Margaret. But it can also shrink because you have forces that delete these elements because they can recombine between each other. Don't apologize for the technical difficulties that happens. So they can recombine, and so it means that everything that is in between can also be deleted by a process of non perfectly targeted recombination. So you get two forces, like expansion of the genome, mostly under these replicating elements, but also reduction, either under selection or because of these non-properly targeted homologous recombination. Some genomes, like the ones of axolotl and other species of batracians, are pretty big because they have been completely taken over by transposons and retrotransposons. So they would consist like 80-95% of retrotransposons, much more than in the human genome. There's a question from Tasneem, I think.
Yeah, hi, Professor. I just wanted to ask about the retroviruses genomes. So basically, retroviruses integrate their genome inside the DNA in order to replicate. But the retroviruses repeats that are here are from ancestors, you mean, right?
Yeah, absolutely. So yeah, that's a good point. Like these are not things that we have acquired during our lifetime. We inherited it from someone who got infected millions of years ago. Like there was the equivalent of HIV at the time, like when we were not even split from monkeys, and one of these monkeys got infected. This infection went to the germline, not just in the soma, and got passed down the generations. And at some point this virus mutated, so like it cannot produce anything infectious anymore, but it's still there in the genome, and that's 8%. That's not small, because we have kept a track of the infections of all our ancestors. We keep tracks of it. It's rare that this goes inside the germline. That happens, and when it does, then we inherited it.
Okay, so mutation stopped the replication.
Yeah, some of these retroviruses are not mutated. Some people are a bit scared, because we have these dormant retroviruses in our genome, and one day they could maybe wake up and then restart.
They might appear, yeah.
But in humans, they're not active. In mouse, you have some of these that are active, like they're inside the genome and they can go out. But in humans, nobody has ever found one that would be able to leave the cell and start a new infection.
Okay, thank you.
Pleasure. So that's about it for the repeats in the human genome. Let's zoom in a little bit on these non-LTR retrotransposons. This is by far the most frequent class in the human genome. So they consist of ORF1 and ORF2. It's kind of 8 kb, 6 to 8 kb. ORF1 has got a it produces a protein that protects the RNA, and ORF2 has got multiple functions. That's the main protein of that type of elements, because it's the reverse transcriptase. That's the one that will convert the RNA into DNA. And that's also the integrase that will put this DNA inside the genome. So the transposon is inserted here in the place that's indicated in red. It will be expressed, so that produces an RNA. That RNA is translated, produces the proteins that immediately attach onto the RNA that were used to produce them. And ORF2 being a reverse transcriptase, it produces a cDNA that's then put back again by ORF2 inside the genome, but at another location. Okay, so the intermediate looks like this, protected by ORF1, and then bound by ORF2 that will produce the cDNA and incorporate it into the genome. Non-LTR retrotransposons are transcribed, translated into a reverse transcriptase, so that's the ORF2, reverse transcribed, and integrated into the genome. The insert usually contains a poly A tail, which is there, as a relic from transcription. So it's a very minimal system. It produces an enzyme that just makes a copy of itself and goes back to the genome. The sign, or the ALU repeats in the case of the human genome, are kind of super parasites because they're parasiting the L1. And they all come from an RNA, which is called 7SL, that has this structure here. It's pretty short, like it's 280 base pairs, has got a poly A tail, and it just happens to be very good at replacing the RNA here, which is usually the one of the line 1. It just goes instead. So the ORF2 thinks that this is its own RNA and makes a copy of it instead. So it's really parasitizing ORF2. So some RNA are very good at catching ORF2 and be reverse transcribed back into the genome. So ORF2 doesn't do any quality control. It just says if there's an RNA that latches onto me, I suppose it's mine, and I'll make a copy of it and I'll put it in the genome. And 7SL just happens to be very good at it. Nobody really knows why. Maybe the abundance or something like that. And so that makes copies of size 280 base pairs, more or less. But they're not autonomous. They've been produced by the ORF2 of line 1. So retrotransposons are super weird, and so there is a connection between retrotransposons and retroviruses. So LTR retrotransposons are transcribed, packed into a capsid, reverse transcribed, and inserted back into the genome. That's the part that is in purple here. So you have a LTR stands for long terminal repeats. That's how you recognize them. They have these something that is around 500 base pairs on the flanks that are exact copies of each other. And so they produce an RNA. This RNA is exported, then translated, and the product of translation is a capsid. So they really make a capsid just like a virus. And another product of this is a reverse transcriptase. So that RNA is then converted into DNA. That capsid is put onto the nuclear pore, injects the DNA inside the cell, inside the nucleus I meant, and that DNA is then integrated at another site of the genome. So you see it's a closed loop. They make some sort of an auto-infection. They cannot leave the cell. Retroviruses have ENV genes for envelope genes. So the only difference is that they're able to coat that capsid and to export it. And then you can imagine that the cycle would continue in another cell. So there is a relationship between retrotransposons and retroviruses, in the sense that a retrotransposon that would acquire ENV genes would turn into a retrovirus and would be able to leave the cell, and vice versa. A retrovirus that loses these genes here will turn into a retrotransposon. So there's a question, so is this how genes are duplicated in a genome? They can, but mostly not. Like this is the way that we keep acquiring new copies of viruses in our genome, and you've seen that there are many. We're talking of hundreds of thousands easily. but the way genes themselves, like functional genes, are duplicated is another mechanism. And usually these are whole genome duplications. Like, you got 10 chromosomes and one day you got 20. So you have effectively doubled the number of chromosomes and number of genes. Somehow the cells live with that, but you have just doubled the amount of DNA. That's the typical way that genes are duplicated. But you also have other mechanisms of duplication. We're going to see this in a minute. So the only difference between retroviruses and retrotransposons is that retroviruses do not leave the cell because they have no envelope genes. But that's kind of weird, right? You'll have to agree with that. Something that just even almost makes it out of the cell and in the last minute just comes back to the genome. That's kind of weird. They're also DNA transposons. It's the product of step 4 and mRNA. Here, that's a completely normal mRNA. So they use polymerase 2, there's a poly A tail, etc. That's a completely standard mRNA. You can look up the mechanism of duplication of the creation of the LTRs here by HIV. This is fascinating. Like how complex that step is, everything that happens here in the capsid, here or there. It's really, really super complicated to go from that poly A tailed mRNA back to something that looks like a virus again. It's like, the number of steps of that reaction is really, really large, and it's quite confusing. It's amazing that nature comes up with such complicated things, but that's a completely normal mRNA there. So DNA transposons, that's the last class of mobile elements in our genome, usually have a cut-paste instead of copy-paste replication cycle. The transposase recognizes the inverted repeats here, so these are repeats that are palindromic, they're pointing opposite direction, but they're not palindromic, they're just pointing opposite direction, and moves the transposon. The transposase will mobilize any element with inverted repeats, so transposons can move even when they are dead. I'll explain that in a second. So the typical architecture of a transposon is these inverted repeats that are the recognition signal and the transposase here. So once the transposase is expressed, it goes back in the genome, in the nucleus, recognizes the inverted repeats, and then cuts whatever is inside and puts it somewhere else. So it cuts there, and that leaves here a little scar. You can see that once this thing is gone here, the excision site still has this little scar, this blue part that belongs to the transposon, and it chooses a new site for it, and it cuts, it doesn't cut blunt, it cuts with a staggered end there, and so that means that you will have duplication of these nucleotides here. This is called target site duplication. You'll see TSD sometimes. So this is replicated just because of the way this was initially cut, and the DNA is repaired like that. And so whatever is here, even if it's a non-functional transposase, there can be another transposase coming from another transposon, another copy of the same transposon to be more accurate, that recognizes these inverted repeats and says, I'm going to cut this thing here and put it somewhere else. So even if that is dead here, it can still move. As long as there's one transposon in the cell that produces the transposase, all the transposons can move. They need to be all dead before they stop moving. So DNA transposons are quite frequent in bacteria, but not so much in eukaryotes. Like bacteria have barely any retrotransposons, and for us it's the reverse. So how is it cutting the sequence? The transposase is an enzyme, it's an endonuclease that is sequence specific. It recognizes the inverted repeats, and because they're inverted like this, it kind of puts them like that. There's this intermediate mechanism where it puts it bundles the DNA based on its sequence like this, and then cuts it, gets both at the same time, something like that. It's because the transposase is actually an endonuclease. Its main job is to cut DNA. So in the non-mobile type of repeats in the genome, there are the satellite repeats and the telomeres. So the satellite repeats are sequences that were found in the centromeres of animals and plants. Centromeres, not telomeres. They were discovered by ultracentrifugation of DNA in sucrose gradients. So back then people could not sequence much or anything actually in the 70s. So the only way they could analyze DNA was by centrifuging it at crazy speed and see where it would go. So you just sonicate the DNA to break it into small pieces, and when you centrifuge it, in something that has more or less the same density as DNA, then you get a band, a fuzzy band here that corresponds to everything in the genome, but you have these satellite bands that correspond to things that have exactly the same sequence all the time. And so they migrate at one particular point in that experiment. And so people could actually get these bands, purify them, and eventually they could sequence them. And so they called it satellite DNA, just because there were these satellite bands there. And these are repeats in the centromeres of human, mouse, etc. They're called satellite repeats, satellite bands. Telomeres are the very ends of the chromosomes. They are formed by 10 to 50 kb of the same short repeats, and that TTAGGG in the case of vertebrates. So here that's the end of the chromosome, and the telomerase here, which is represented by that yellow enzyme, elongates the chromosome always by putting the same repeat here, TTAGGG, in the 3' direction. You can see that here it elongates it toward the right, which leaves more space for Okazaki fragments here, this primer, to bind and then start the replication of the chromosome there. If you didn't do that, it means that every time the Okazaki fragments are losing a little bit of space, they cannot really start at exactly the 3' end of the chromosome. They have to always be a little bit inside. So that's a way to make sure that you always have a bit of a buffer so that you can go through replication. When the telomeres are gone, because you have replicated them away, then the chromosomes can divide, and the cells just can't divide as well. The chromosomes can't be replicated, and the cells cannot divide anymore. So that's another type of repeat. This one is very simple, TTAGGG, and that is present hundreds of thousands of times, millions of times in our genome at the ends of the chromosome. That's the telomeres. Another kind of repeat that will be important in the coming classes are the repeated triplets. Repeated triplets are unstable, which means that you can increase the number of triplets or decrease the number of triplets. They tend to either increase or decrease in numbers. This causes a large degree of polymorphism in so-called VNTRs, the variable number tandem repeats. VNTRs are used for genetic maps and forensic analysis. So the mechanism is this one. You have this very low complexity sequence, CTG, 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 etc. Can be a large number of times. And during replication, the strand in blue at the bottom is the template, and the one in red is the one that is being elongated. Imagine that while this happens, the strand in blue kind of, loops over and anneals directly to the end like that. And that's possible because the sequence here is perfectly identical to that. You just loop out a couple of the nucleotides in blue, and then you have already a perfect matching. And that can be missed by the DNA polymerase, and it thinks that it has replicated everything, but in reality it has missed 2 or 3 copies here that are in the loop. That's how you can lose copies during the replication. You can also gain copies if the loop is happening on the side of the red, not on the side of the blue. In that case, you're gaining copies. And that happens not a lot, but that happens enough that anytime there are triplets that are repeated, like CTT, 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 most of us will have different number of copies, because in our parents and our ancestors, these mechanisms have happened, and they've either gained or lost 1 copy of the triplet there. And so that's very useful to create fingerprints, like we're going to see, for genetic maps and forensic analysis. Here, that's also an example that we've seen before, where you have a constant sequence on the sides, but inside, the number of triplets CTT is different between every individual. So some genes are properly repeated, and this is where we get back to it. So most vertebrate genes have paralogs. These are genes that are homologous together within a genome. That's a paralog. So you got twice the same gene, but they're kind of a little bit different. Their identity can be like 80%, 90% at DNA level, but not 100%. But some genes have multiple exact copies. Like there's really exactly multiple copies of that gene, and this is the case of the histone genes, the tRNA genes, and the rRNA genes. So like the DNA copies of those genes or in tandem. You have multiple copies, one after the other. So how did that happen? I'm not sure, but this is definitely the product of selection. So that happened for a random reason, and that was kept because it was good. The products of those genes are among the most abundant in the cell. So the number of histones you need, of molecules of histones you need in a cell is really huge. So that's the way to boost the production. You just have multiple factories that produce the RNAs. You know, each gene is an RNA factory. And if you have multiple copies of this gene, you have more copies of the RNA, and then you have more protein at the end. So the products of those genes are among the most abundant in the cell. Repetition allows the production to be boosted. So that's the reason. So these are exact copies. They're not just paralogs. Is you have actually several copies of the same gene, and they come in tandem. They're really close to each other most of the time. But you also have other copies at other places in the genome, and I don't know how this happens. Actually, it's a good question. Maybe it's known. So that's it for the description of repeats in the genome. Now we're going to focus a little bit on the method. How do you study these repeats, and why does that matter that they're repeats? So repeats are Achilles heel of genomics because they cause all sorts of problems. Repeated sequences are a challenge for mapping and for assembly. For mapping, it is impossible to know which loci the reads are from, and for assembly, several genome versions are compatible with the reads. So think about it. You have a read here represented by that little line, and the repeats, it's like twice the same sequence, exactly the same sequence in the genome. If your read has that sequence, you won't know whether it actually comes from that location or whether it comes from that location, and you can't know. Like, these sequences are exactly identical, so you can't decide that. If you're assembling a genome and then you have these tiling reads that you're trying to find overlaps, the problem is that you cannot distinguish this assembly from that assembly. Okay, if you look at it, the reads that are members of that assembly here are exactly the same as the reads that are the members of that assembly. You have the green reads, like the dark green reads. You have the light green reads, and they correspond to unique sequences. They're easy to assemble. But if you have multiple tandem copies here of that repeat in the middle, all you have are these purplish reads, and you cannot distinguish from, let's say, a scenario where you have 2 copies or 4 copies, or actually any number of copies. You know that there are at least 2 copies because you have the reads that are in between the 2 copies, but you don't know how many copies there are. So that's a bit of a problem. If you have repeats in a genome, you can't assemble them very easily. So one thing that's quite important for you at that stage is to be able to understand the differences between the read is mapped uniquely, the read is not mapped at all, and the read is mapped in a repeat, and therefore I don't really know where it is. So you can recognize here the SAM format. I'm showing you an example from a real mapped read. You have the header of the SAM format here, and you have 3 examples of reads. One that is called unique, another one that's called repeat, and the other one that's called unmapped. And if the read is unique, you will have a location, like 2L at that particular location. Pay attention to the map inequality, it's 60, so it's very high. And then you would have 0 errors if you look at the last fields that are at the very end on the far end of the line. A pretty high score and no errors. If the read is mapped in repeats, it will have a location. Okay, it says 3L position 2305, okay, but the mapping quality is 0. It says, I think it goes there, but in reality, it could go somewhere else. That's really the meaning of mapping quality here. So don't get confused. Both are mapped, but one has got high mapping quality, the other one has very low mapping quality. And there's no errors. Okay, the problem is not that there are errors, it's just that there is another sequence in the genome that would match perfectly. That's the definition of kind of mapped in repeat or poorly mapped read. And then you have the read that is unmapped. It means like it doesn't go anywhere. That sequence was not found in the genome. In that case, you don't have a location. There's a star here for the chromosome and nothing for the position. There's also no information about the errors, etc., because there was never a match. So don't get confused between no match and mapped in a repeat, which means several matches. So reads and repeats have a defined location, but a very low mapping quality score, MapQ. they typically have a low number of errors, indicated here on the last fields, and a high alignment score, also AS stands for alignment score. Reads in unique sequences have a high mapping quality, 60. Unmapped reads have no defined location and also very low mapping quality. So bear that in mind, mapping quality 0, you can still have a defined location, it's just that this location is not unique. So why would anyone assemble a genome, like we're going to do a little bit later? Genome assembly is not only to establish a reference genome, like it was done in the case of the human genome, it can also be used in metagenomics to take a census of bacterial populations, or even to discover new organisms. You just, sequence the guts of someone, or you sequence dirt or underwater, like water, very deep, and then you want to assemble the genome of microorganisms to know what they are. It can also be used to obtain the genome of individuals, especially at complex variable loci. For example, the HLA gene. Sometimes you would actually reassemble locally the genome of individuals because our organization of that locus would be different between people. So the simplest is just to reassemble it from scratch every time. So there are many reasons why you don't assemble the genome just once. That's not something we do just once for the human genome. Like you would have genome assembly tasks that are still ongoing today. So as you know, we've described in the first class the way that the human genome was sequenced by the public consortium. But there was the one that we did not describe, which was the one of the Cellular Corporation. And that's the way that most genomes are assembled nowadays with that strategy, because it's much cheaper. And that's what we're going to describe in a little bit more details right now. So you start by doing a shotgun sequencing of short insert clones, let's say 1 kb, 2 kb, shatter the genome in many of these small sequences, and sequence the hell out of them. And if you sequence the same sequence twice, it's fine. Like you don't really care about this. You just want to have a lot of references of these short sequences. You also get longer sequences. In that case, that'd be 100 kb, and you sequence the ends of these 100 kb. They're called paired end sequencing. You're not sequencing the inside, but you know that they are within the same fragment, 100 kb of each other, and you have the sequences at this side. You make contigs with the small reads, the ones that are in orange, like they overlap, so you can make these contigs, and you use the green ones to make scaffolds that you link the contigs together, and you use the sequences that you've obtained at the ends to be able to bridge them in a sense. So that's the general strategy of shotgun assembly that everybody uses today. So when this came out, the major innovation was not really the sequencing technology, but the assembling technology. So that was a real revolution when that came out, and this was the cellular assembler. And that's here something that I'm not going to go through because it's not that important, but just for you to understand that an assembler is not just something that puts the reads next to each other. It's actually a very complicated program that takes many decisions and that trims out the reads, removes some, puts them in the trustable category, some others in the not so trustable category. So the diagram of operation of an assembler is not straightforward. It's actually a very, very difficult problem, and it's a feat of engineering for people who want to do that. So the cellular assembler, and we're talking end of the 90s, early 2000s, introduced the concept of a unitig. And a unitig is a high confidence contig. So it's a contig that you're pretty sure is correct. Repeats were masked at an early stage of the project and reintroduced later to build contigs. So the real problem of assembly is repeats. And why was the cellular genomics endeavor able to come up with a pretty good version of the human genome? Is because they addressed that problem relatively well. And the key for them was this idea of a unitig. And this diagram here shows how they assembled the contigs in the scaffold. It's basically, to cut a long story short, that they had different levels of trust in their contigs and in their reads. So the unitigs were 100% sure, so they were super easy to put next to each other. But they also had lower confidence contigs that they called rocks and pebbles. And based on that, they kind of made a plausible association of the contigs. And they were sure that they were bridging them by things that were high confidence. And based on that, they could scaffold the contigs next to each other with relatively good quality at the end. So this strategy is called overlap layout consensus. And so the overlap graph shows which reads can be stitched together. So the idea is that you're trying to put reads next to each other. You say, okay, I've got a read, I've got another read, can I kind of, is the end of this one the same as the beginning of the other one? Can I use them to make an overlap? That's really the strategy that was used at the time. So they made an overlap graph looking like that, showing which reads can be stitched together. Each node represents a different read. The layout step is to find a Hamiltonian path, so that is to say a path that visits every node exactly once. In order to assemble the genome, you need to use all the reads that you've got and use them exactly once. You say, I cannot leave any read out. Okay, like every read comes from the human genome, so it has to go somewhere, and I cannot use a read more than once. So that's the problem you're trying to solve here. And to find overlaps, one needs to align every read with all the other reads. Okay, to be able to say, this read goes there, that's a hard problem. Like the main issue with this strategy of the cellular genomics cellular assembler, is that it was inefficient because of that. It works, but you needed really tons and tons and tons of computing power, and in the end, not everybody has that, of course. So then came a pretty big revolution in the early 2000s, is to say we need to assemble the genomes in a different way. Like same sequencing technology, but we're changing the algorithmic background. And we're going to use the De Bruijn graphs. I'm not sure how to pronounce this. This is Dutch, and I think it's De Bruijn, but I'm not sure exactly how to pronounce that every time. So the idea is the same, but not the same. So the De Bruijn graph shows which k-mers you, from the reads can follow each other. So you're taking your reads and you chop them into k-mers. Remember, these are the sliding windows text that you have in DNA. And instead of making a graph between the reads, you make a graph between k-mers. So each node represents a different k-mer, and the assembly is a Eulerian path, that is to say a path that visits every edge exactly once. And that seems stupid, say it like that. Say you go from a problem in which you have to use every node exactly once to a problem where you have to use every edge exactly once. But the difference is that the second problem is easy and the first is really hard. So by just doing this, the person who invented that really reduced a lot the computational needs to be able to assemble a genome. So that was a pretty big revolution when that came out. So building the graph does not require any comparison between the reads. That's the thing as well. Is that you no longer need to say this reads overlaps with this one. You just take the k-mers, put all the k-mers in a bag and say, after this k-mer, which are the possible k-mers that can go, by just shifting them by one, like this is represented here, and which are the ones that I have, and I'm going to find a path with all the k-mers that I've sequenced. And if I can do this, then I can assemble my genome. So here you have an example of how that would play out with the reads that are here, and there is a sequencing error that is here in red, and the node says the graph here in 2 has no Eulerian path, so it's not possible to assemble the genome here because of that sequencing error. So you also have a way to know, okay, there are sequencing errors because there's no solution to the problem. So it's fairly complicated. It's okay if it's hard for you to follow right now. This is something that you typically need a little bit of time to appreciate. But just to say that there was a very big conceptual revolution when they understood that you can go from a previous strategy of overlap layout graph to the De Bruijn graph, which solves the problem with much, much less computer power. So that doesn't solve everything. The repeat problem persists. So you have a genome that looks like this, for instance. The reads would look like the ones that are here. So the De Bruijn graph does not always have an Eulerian path. So we've seen a case in the previous slide where there is no Eulerian path. One often needs to add edges to create such a path. So if we take the example here, you take the k-mers that are represented by these circles there, and you'd be able to find unique linking between the k-mers. And there is a path that crosses everything like this, but because you have multiple times the k-mers of that repeat going into the k-mer of the beginning of the repeat, then there are edges that go like that. Okay, so there is no way to make to traverse this whole graph by taking every arrow exactly once. Okay, to assemble the genome, you need to find a path that goes through every arrow exactly once, but there's no solution here. So what you need to do is to put more arrows, to infer that there must be more arrows that are here, and then you have such a path. And the path is you go like this, then you take this, and then you go like this once again using the other line of arrows, and then you can finish it. So you've gone through the repeat twice. So that's the way you solve the problem. But if you had 4 repeats, not just twice, not just 2, then you're missing 2 repeats. You haven't discovered them at all with that method. That's what I mean. So this is the so-called route inspection problem. You can just add these extra edges and then find a new path in that graph. But in general, repeats are still poorly resolved at the end. You go from this to only 2 repeats. You haven't completely solved the problem, even though you have been able to go through it in much less time than with the previous method. So for this, you need scaffolding, and paired and reads are really useful. Paired and reads allow contigs to be linked together, but the space between contigs is unknown because the length of the paired end fragment is unknown. Okay, so you got the small reads at the top. That's the one that we represented in orange in the cellular strategy. But you also have these super long reads of which you know the end. And these are the ones that were represented in cyan, like blue, in the cellular strategy. And so if you have a read like this that has an end which is in dark green, another one which is in pale green, you know that dark green must be not so far from pale green, right? And if you know that the approximate distance here is 100 kb, you know that the distance here must be 100 kb as well. The issue though is that you don't really know how many repeats there are. You don't know the exact size of that fragment. So you have to assemble the genome like that, to say there is a contig here on the left in dark green, another one in pale green here, and I know that they're next to each other and that the distance is approximately 100 kb, but I still have a gap. I don't know exactly the sequence that's there because I don't know the number of repeats exactly. All right, I hope that this is clear, that this helps to have these long reads here with the ends, but it doesn't really solve the problem of knowing how many repeats there are there. So finally, what does help, what needs to be done? There are long reads. Long reads can go over repeats, over repeated regions, making the assembly much easier. Okay, if you sequence the whole thing now, well then you know. Like if your read has been like this, then it's trivial to say there are 4 repeats because I have sequenced 4 repeats. It's as simple as that. So the longer the reads, the easier the genome assembly. However, the error rate is approximately 10%, if you remember, even in the case of the nanopore technology and the other one, I just forgot the name right now, of the other technology. So they have approximately 10% error rate. So that's the problem. You would have a good assembly, but the nucleotides, the SNPs, or whichever nucleotides you have, would be mostly wrong. Not mostly, but 10%. So you have hybrid assemblies. So they combine short reads with low error rates and long reads with high error rates. So you do both, and then you get the best of both worlds. You've got the long reads that will allow you to count the repeats, and you have a good layout for the genome. And then you use short reads that will tell you exactly what's the nucleotide everywhere. The combination is equivalent to having long reads with low error rates. So the technology I was looking for is PacBio, Pacific Bioscience, of course, the other one that is long reads with 10%. And there's yet another option, which is to not use sequencing at all. And this is called optical mapping. And this is actually used relatively frequently. People have these technologies when they sequence genome. And optical mapping consists in labeling chromosomes at specific sequences. Let's say you would take 6 nucleotides, GAATTC for instance, and every time you have that GAATTC, you have a way to really mark it with a fluorescent label on the chromosomes. So you take the chromosomes, you kind of put them with a very sensitive technologies. You can put them on a glass slide, like they're all completely parallel and elongated to the max extension. And every time you have that sequence, there would be this little green dot, a fluorescent dot that says right here there is a GAATTC, and you find them. And then you get kind of a Morse code, like dot dot, dash dot dot, etc. and that tells you, okay, in the genome, the distance between the GAA-TTC sites would be exactly like that. You know the distance because you have elongated the chromosome to its max extension. And based on what you sequence, then you have to match these high-level markers, okay? Like you propose an assembly and you will see whether, okay, do I get exactly the same optical map, like when I compute it in vitro, in silico I meant, when I compute it in silico, do I get exactly the same pattern of representation of these green dots as what I obtain experimentally? And you can have gain or losses. If you see that you're losing stuff here, well, you have to assume that there has to be a sequence there in the middle, and you can look for your reads to see what that could be. You can also see that something has been duplicated compared to the experiments to say my assembly is wrong because it assumes that this sequence is present 2 times, but it should be present only 1 time, etc. I mean, you see the idea, right? The exact spacing between these dots makes kind of a fingerprint, and you can look for this fingerprint and match it with your sequence to make sure that your fingerprint matches the experimental data that you got this way. So it's a very cool way to make sure that the assembly is good and to detect errors in the assembly. So that's it. That's a pretty dense part, and most of the details are not super important. What matters is the practice. But the key points to remember from this theoretical overview is that repeats are the major problem of genome assembly. That's the one thing that makes it hard. Otherwise, it'd be pretty easy. And most repeats in the genomes that we manipulate every day are mobile elements. They correspond to like 50 or even more % of the genome in the case of humans. So now this will be time for a break, and after we're back from the break, we'll do this exercise. So let me read the exercise to make sure that people kind of understand what we're going to do. Then we have a break, and you'll do this in the breakout rooms. So we sequence the mystery bacteria, and we want to assemble the genome to know what it is. We will use the assembler spades, that stands for St. Petersburg assembler. It's made by the guy who invented this approach with the brain graph. A pretty smart guy. So first connect to the teach cluster. You can do this right now if you haven't done so, and follow the instructions below. So copy mystery_1 fastq.gz and mystery_2 fastq.gz from my home. Copy the serum script from my home. You need to edit the script to be able to run it. There are instructions inside that will tell you what you have to do. You'll see it's not very hard. It's just to make sure that you get practice with that. And then run the script. It should be approximately 10 minutes of run with the way I configured it. And that will assemble the mystery species. Then you need to answer those questions. So which line of the Slurm script speeds the job by a factor 8? How is this achieved? In contigs.fasta, the contigs are sorted by decreasing length. How big is the largest contig? How big is the smallest contig? And you have a hint for how to solve that. How many contigs are in the assembly? And finally, of course, the only question that matters, what is the mystery species? And you have a couple of hints to help you solve that, because it's not always easy to manipulate the data when it's on the cluster. So I'm going to assign you right now in your breakout rooms. We have to get started. Let's say take a 5-minute break, but we want to be actually working together from 10 past. So at 3:10, I would like everybody to be together working on that. And what I will do is I'll give you a 10-minute head start, and after this I'll be touring in the breakout groups to make sure that you guys are making progress and to answer your questions if you have any. And depending on what I see and the progress that you guys make, then at some point we're going to come back together and discuss the answers, and we're going to also hopefully record the way that we can solve these things. So that's it. I'm going to put you in breakout groups right now. I'll stop the recording now. Yeah. So welcome back. The recording has restarted. So let's see how we can solve these problems here. All right, can you guys see my screen? Yeah, okay, that's good. So here on this window here, I'm already logged in on the cluster. So if I do pwd present working directory, I see that I'm on Scratch. So that's my Scratch space. You must be really careful that in the beginning you do cd $scratch. If you make a typo to it, or if you write it in a different way, then you will go home instead and it won't complain. So you have to be a little bit careful with that. So I have copied mystery 1, mystery 2. If you have looked at the files, they're paired end. They're actually from the same experiment. And the let's have a look at the spades slurm script that is there. There is this new line here that has been added compared to the previous types of files that we have seen, and that requests 8 computers, 8 CPUs for each student. And so this is why it goes 8 times faster. It's just because I'm requesting 8 times more resources, compute resource, than the number of tasks. So then you can have 8 computers doing the assembly on their side and then creating everything together at the end. So that's the line here that speeds it up by a factor 8. If you don't put this line, then you have to wait substantially more than 10 minutes. So that's it. Sometimes it's a good trick to know that you can speed up some computations this way, but not all. In that case, that works. But some others, it won't really speed it up. Then you have to replace X and Y by these files. I think everybody got that point here. You have to put mystery 1, mystery 2 here. So that's the first file, that's the second file, and everything will be in the spades underscore output. So I think everybody managed one way or another to go that far. Then that creates this directory here, spades underscore outputs, and spades output. And there, it's a pretty long listing. There is the file that is called context.fasta. When this file is there, then the run is finished. So if we use less to see that thing here, we could see that the name here of the sequence is node 1 and length is that. So the length is 251,000 and then 19 base pairs. Okay, so that's the length here. The coverage, 12. So this thing has been sequenced 12 times, more or less. So that's a pretty high coverage, which means every nucleotide was in 12 reads, you know, like that stacked to sequence that nucleotide. So we've got good confidence. So that's the length here. That's the node 1. There is this command minus capital N unless, which shows the line number. You can see that this is the beginning of the file here, line 1. And if I press capital G, I go to the end of the file. So the last line is 82,451. And this bit here is the last config, and because they're sorted by length, the smallest has length 128, pretty small. So that's the length of the reads, I think. It's just one read, there's almost nothing. And this is config number 243. So there are 243 configs. How did you show the list? Sorry, I missed it. So I'm not sure which list you're talking about, Gloria. So if you want to show the row numbers that are here, you don't need them. It's just for me to highlight that this is the end of the file. It's capital minus capital N, and with small g you go to the beginning of the file, and capital G you go to the end of the file. So you can quickly teleport to the top and to the bottom. Things are more complicated when you want to go to the middle of the file. Then you have to scroll with F for forward and B for backward. So, all right.
Sorry, Professor. I just
Yes.
meant, how were we able to see the numbers? I just looked away for a second and I missed the command that you did to show like all the numbers on the side. Yeah.
You don't need them, but if you want them, it's minus capital N.
Okay, thank you.
Minus capital N, like that, inside less. So G, like small g. Oh, damn, it autocorrects with capital G. I can't even type what I want in the chat. So small g for top, capital G for bottom. Got no idea why small g for top, capital G for bottom, but that's the way it is. Okay, so that answers these questions. They were not the hardest, so that's pretty nice. You can see that the assembly has worked nicely. Let's go to the top. So now if I want to know what is this mystery species? Well, it's quite simple. I just take that here, and on my Mac, I press Command C. I've made a beautiful copy of it. And then all I need to do is to go to the browser, to Blast, and then to paste it in Blast. Okay, so that's the way you can solve that problem. You can just, if you have a Mac, if you are able to copy paste things from your terminal directly into your other computer, like your computer at home, then you're good to go. You can paste that in Blast, and that will be enough to identify the species. If you can't copy, then you have to retrieve the file on your computer where you can edit it and copy it. So that's why I give you these hints. Okay, so this is not on the cluster. Now you need to open a new terminal or to close this one. So I've got this thing here which is on my Mac and BIOD25, and you can see that this is not a path on the cluster. This is a path on my computer at home. And the command would look something like that. SCP for secure copy, and then you log in, and then here the computer, the teach computer that we're using, teach01, and then the path of the file you want to copy. Okay, so because I'm copying directly from scratch here, this is the whole path, like // and the whole thing. If you the default path is home, so you can just copy this thing in home. So you can do copy context FASTA to my home here, and if I do ls of my home directory, I see that the file is there. Okay, like that's my home here on the cluster, context FASTA is there. So that would allow me to simplify this command a lot, because by default everything is supposed to be in home. So I can do that. I can try to copy this file from my home on the cluster to BIOD25 where I'm here right now. So if I press enter, it asks for my password. So I enter my password here, and you can see it works like that. It has appeared here on this window that you guys can see. And then I can I could double click it, but my Mac is going to complain about the FASTA extension. So I say, dude, it's just text, come on. Yeah, that's text. And now I can double click it. And I've got, a good old graphical interface. I can use my mouse to do all these things. I can copy, I can paste it, and that's why it's cool. Also, what's quite interesting is that I can delete everything else except node 1, or I can maybe even create a new file here which contains a bunch of node 1. I copy, I paste, and then I can use that as the input for Blast, because Blast allows me to use files, not just to type in. Okay, did anyone manage to blast this first node 1, this thing here? E. coli! Excellent, very good. So that was the very mysterious species. That was actually the first bacterium that was sequenced. That's of course E. coli. It was not that difficult. You get 100% match. This is really like a total match. So that's the way you can solve these exercises with a bit of computer cluster. Should have known, of course, should have known it was E. coli. It would not be anthrax or something like that. So a bit on the cluster, and if you can copy paste things directly into Blast, that's great. If you cannot, you can try the SCP. Has anyone managed to do this SCP trick that I showed at the end? Oh, it's not working on you guys' computers, because I've heard from some students that it was not working. It worked for me. Okay, one person managed to make it work. So in principle that works. I tested it on MobiXterm, but I was at the university, and so I don't know if there are some restrictions, like some firewall would prevent it from working, etc. If you guys have difficulties with it, like you can write to me or to Michelle, we'll try to help you, but in principle that should work. Did not find the file that was copied. Yeah, exactly. Another difficulty is that if you're using MobiXterm, okay, so the file here has been put into my BIOD25, but it's because I know where I am on my Mac. If you're using MobiXterm, like you don't really know where you are. But after you've done that line here, you can do ls, that should show the file on your MobiXterm, interestingly, you can do PWD that will show you where you are in MobileXterm. And what you can do, which is pretty neat, is to say I want to make a copy on the desktop because there you're going to find it. So you do CP context.text. Now it's called context.text because I changed the name on desktop. Okay, so if you're using MobileXterm, the desktop will be directly accessible from where you are, and so that will make it appear on the desktop, and it's going to be super easy to find when it's there. That's the trick you can do. So in that case, on my Mac I can do that, and you see it appears on my desktop. I cannot miss it there, but on Windows it's going to be in a place that's not trivial to find. So my advice is copy it to the desktop, you're going to find it immediately. Alright, so SCP context FASTA, no such file or directory. Yeah, no, because that's not what you have to do. That's not the way you have to to make the command. Here I can do it this way only because I have made the copy from scratch to home, because here after the colon it assumes that this is on my home. If I haven't done that, then it's going to say, yeah, I can't find it. So make sure you copy it home first so that it simplifies that line. Like, of course there's a million chances to make a typo when you type this, so that's why it's kind of a little difficult. I'm going to stop sharing my screen now and answer any questions that you may have. I think Theodore and Tazit, are you saying goodbye or you want to ask a question?
We're gonna get going. If there's any questions, when are you in your office next?
There's an office hour tomorrow, and after this I'm gone. Like for reading week I won't be around. I'll be answering my emails, I'll be working, but I won't be physically in my office.
What, are you leaving Canada?
No, I'm going to Quebec.
Oh, hey, me, hey, that's awesome. Guess well,
have a good break at Quebec.
Yeah. Maybe some people have actual questions, so let me ask, because now time's running out. So if people have actual questions about what we saw together, or things about the way to do the midterm, now is the time. Anyone want to set up a Discord? Yeah, guys, work together. Like now is the time. This is one case where I want you to work together. I want you to share the knowledge that you've acquired. This is something that's kind of new for most of you, so don't be shy. Work together. My advice still is be still careful with whom you share the information with and how much you help other people. You know, there is this saying, give a man a fish, you feed him for a day. Teach a man to fish, you feed him for a lifetime. So what I would like is you to learn from each other, not to get the answers from each other. So try to be responsible. I trust you. Of course, you don't have to be responsible. I cannot control you. But I would like this to be a teaching channel, not a getting the answers channel. But you guys are adults, you do whatever you want. Like, not related to the cluster, but what is the origin of line and signs? Oh, such a good question. Very good. You have the Discord. I'll post the Discord on if that's okay with you, Wensu. Can I post the address of the Discord on Quarkus? I'll post the link. The origin of line and sign, it's like sign is pretty clear. Okay, it comes from the lines. Like this RNA just found a way to be reverse transcribed by the ORF2 of line. That's fine. Where do lines come from? Very big mystery. One thing is viruses were probably from the beginning of life, actually. Like you can really trace them until the absolute beginning. And so lines must have been some kind of ancestor of the of these retrotransposons at the time when really the reverse transcriptase was just born, in a sense. And so what I explained with the retrotransposons, the LTR retrotransposon retroviruses, is that, you know, they keep oscillating between virus and transposon phase. So sometimes they live as a virus, so they can go between cells, but then they lose the envelope, and so they become transposons again. But then they would reacquire from another virus, the capacity to go to another cell. But these are, of course, things that happen over millions of years, even hundreds of millions of years. And for the line one, it's probably what happens. Like it's some kind of thing that probably has the capacity to go between species at some point, but loses whatever it needs to be able to go between cells, and then it's trapped within the genome and just makes local copies. But people are mostly speculating about this. Any other question?
Hello, Professor. I had a question about the Achilles heel of genomics. When it comes to mapping, you said that you might have two reads, so you're not sure whether it's whether it comes before the other one, so you don't know the direction. How is that a problem to us? If the sequence is if they're the same, why would we care which one comes before the other one?
That's a very good question. So imagine that what your reads are RNA-seq. Okay, you've got two genes, two exact copies of the same gene, and so they would produce exactly the same RNA, and they would produce exactly the same protein. So why would you care? It's a very good question. And the answer is, imagine that you're interested in something that's not transcription. You're interested in epigenetics. You want to know, for instance, the histone marks or the nature of the histones around the copy on chromosome 1 versus chromosome 3. In that case, you're actually talking of different chromosomes, and you'd like to know whether that particular feature of histones is present on chromosome 1 versus chromosome 3, but you can't. You can't even know whether what you're watching now is coming from chromosome 1 or chromosome 3. And the same if you're interested in knowing the promoters, the property of the promoters, because the promoters may not be exactly the same for these two histone copies. Okay, that actually is the case. They're not exactly the same. So which of these two promoters is active? And you don't know, because you cannot distinguish the expression from gene 1 versus the expression from gene 2. But in general, that's a very valid question. Do we really care? And there are many cases when no. Like we know it's any of these copies that is expressed. It doesn't matter which. But sometimes we do. Does that answer your question?
Okay. Yeah, 100%. I just had a question about you said you don't know whether it came from chromosome 2 or chromosome 1, for example. I did not understand that part because I believe that the two sequences were on the same chromosome, no?
No, they can be on different chromosomes, especially if you're the line, and things like this that are exact copies of each other, but they are on different chromosomes. So it doesn't have to be on the same chromosome. Even in the case of the histones, you have tandems, genes that are next to each other on the same chromosomes, but you also have copies of histones on different chromosomes.
Okay, all good. Thank you very much. Thank you.
Pleasure.
Bye-bye.
Right, enjoy your respite a little bit, so you have a bit of time to rest and to do the midterm. My advice is don't start too late with the midterm, because if there are some technical difficulties, you want to have time to solve them and to ask around. I will post the Discord link right now so that you guys can connect and help each other with that. In the meantime, enjoy and see you in the real world next time, I hope.